We're now going to go on to the next talk, which is absolutely wonderful to have industry now taking an interest here. And Mark Jones, who's going to give the next talk, is from UCB, which is a wonderful Belgian company, which I got to know very well through the antihistamine world, when it really was almost first in with some of the early antihistamines. But of course, it merged with some other pharmaceutical companies, particularly one in the UK, and is now one of the leading pharmaceutical companies in the world. And Mark is somebody who's obviously interested in telling us a little bit about how we, as a research community, might be able to engage industry to develop new diagnostic tests and treatments, which is really why we're at this meeting here. So thank you very much indeed, Mark. It's terrific.
Thank you. So thank you very much for the invite. I've been involved in the CMRC, been a real privilege working with folk like Stephen, Sonia and Chris for the last few years. And yeah, I guess my engagement with ME began probably about 2012. So we've been living and working in Belgium. We came back to the UK. As you can tell, I'm not Belgium. We came back to live in the south, having been up north before. And my son and I used to play tennis regularly. And we'd go one Saturday, and Ben said, not feeling so good today, Dad. You know, maybe we could make it a bit shorter. Essentially, a week after that, he was falling asleep in maths, his favourite subject. And suffice to say, within a month, he was entirely bedbound and was so for about 2.5 years with ME. Eventually, we got a diagnosis. And I guess if you'd have asked me before that what I thought of ME, I'm pretty well ignorant. I've worked in the immunology field for many years. Worked in a company that works a lot in neurology as well. But really, ME hasn't come up very much. The ability for us to latch on to various pieces of molecular work has been difficult. And suffice to say, actually having a son with ME and then subsequently a daughter with ME, so I have 2 kids with ME. The great news is, though my daughter's still, we tend to use it hours, so she's 22 hours a day in bed, was 23, so 1 hour is brilliant. But we delivered my son at University of Sussex on Sunday to do a degree, so that's a real good success story. He's sort of operating about 80% now, so that's really great. So, yeah, and I guess where this title came from is from my daughter, who, being 16 years old and very informed, is quite challenging to me at times as to, Dad, why on earth are you not doing anything for us? Well, you know, what are you really doing? You know, and so this really is a title that was inspired by her. And my talk will actually be in 2 halves. You'll have me talking to you for about 10 minutes. Then we're going to show you a movie, a short film, excuse me, English. It's a film we'll be showing, and I'll talk to you a little bit about that as we get there. But to really try and challenge what it is that we need to do to engage, I thought what we'd do is we'd actually look at 3 areas, and as it happens, they all begin with P. That wasn't deliberate to have a alliteration, but a place to start. Now, this is probably the most important, okay? And I'm going to show you a map here. Now, you're not going to look at the detail, see the detail there. I think it's very beautiful. Does anybody know what that is? Anybody in the audience? Yeah, it's basically the map of how we understand metabolism in the human being, okay? And those of you with great eyesight might be able to spot that in the middle you've got the citric acid cycle, urea cycle, etc. So I put that up on the board here because if you're trying to say, well, maybe ME has some elements of metabolism in it, well, where exactly do we start in all of this, okay? And essentially, what we need to do is to narrow it down, okay? It's very obvious that we need to do that. And we need to narrow it down in such a way that we can actually say, right, If there is some sort of mutation around this particular protein or enzyme or whatever it might be, maybe there's a genetic link there. And we've shown that with the disease. Well, what would that mean in the patients? Well, it may mean that this particular protein goes up or that particular metabolite goes down. Can we go to patients now and see that? Can we actually begin to build the picture as to how this particular part might be fundamental in the disease? And then we have the issue of, well, is that cause or is that effect? So as a virus created it 10 years ago, and we're now just seeing the metabolic effects. So all these questions are really important to us. And I guess, excuse me, this is a real-life example where we've gone from complexity to simplicity. And just to really to highlight what the challenges are. So this is a simple pathway. Excuse me for getting loud. A simple pathway. And I guess the best way to say it is cells need to talk to each other, and they do that very often by having these things called receptors on the surface. So one cell can talk to another and can hear signals to perhaps to grow, to die, to do an immune response. And this is actually a real-life example where in rheumatoid arthritis, which is a disease area I've been working in, we see this particular protein, AKT, is upregulated. That's really exciting. And that leads to an increased activity of autoimmune cells. Again, isn't that exciting? And what we also find out is that if we knock that out, we get a reduced immune response and we get an improvement in joint function, increased mobility. Now, do you think that would make a good disease target for a new medicine? What would you think? Do you think that sounds like a good place to start? Certainly. It sounds like this is actually a pathway that might actually inhibit some of these symptoms that we need to inhibit. Is it going to make a new medicine? And I love this. It's got nothing to do with biology, it's to do with particle physics, but I love this. There are various challenges when you actually try and break things down to the fundamental level, okay? This is essentially a particle physicist. As we know, they basically live in the world of smashing things to pieces and then seeing what happens. And as you can see, it doesn't work particularly well in understanding how a frog works. Though, having lived in Belgium, I'm quite interested in the frog's legs there, but we won't go there. Essentially, there is a problem if you try and get down to too much reductionism, right down to this sort of a level, are you ignoring the bigger picture? So essentially what we're doing here is we look at the bigger picture, go in narrow, and say this seems to be involved in disease, and then come out again. And for our example we had with PI3K, yes, we've done that, and essentially what we see is that that protein is involved in a whole range of different activities. It does a whole range of different things in the body. And actually PI3K is actually a really good target for cancer. So it's a very good anti-cancer target for the various reasons that it controls so many different things. Benefit versus risk is always the question we have. So most of our candidate medicines, they fail for two reasons. One is that you get compensatory mechanisms, which means that that pathway can be sidetracked by something else, and actually you block it, but actually the effects still occur. Or you can have unacceptable effects and side effects. And those two are the main barriers, really, to bringing forward new medicines. You've got to really demonstrate that you really are a very clean target in terms of hitting the mechanism and the side effects. And that's really critical for what we're trying to do. But we need a place to start. And this is one of the most important messages from this conference. We've already had some fantastic presentations we will do, looking through places to start, the story around miRNA, the story around metabolism, the story around genetics that's coming forward. These are great places to start. And what we need is investment in this area, not only in industry, but also in the public sector, in universities, and with folk like the MRC to actually bring investment into this area to help us find a place to start. The other P, the next P, is a pathway to patients. Okay, what we'd ideally like is to have the researcher or drug developer, and this could be somebody in pharmaceutical, or it could be many of the universities now are actually very much driven by the fact that research needs to lead to an end product, and they're very much similar to the way that we would operate these days. Basically, you'd want to have the patient speaking to the researcher and actually saying, these are the key symptoms that are important to me. What can you do in terms of medicines? And one of the reasons why this is important is we did something in the rheumatoid arthritis areas that we asked clinicians what was the most important thing that you thought we needed to do for patients. And so we had 5 things that came out of that list. Essentially, at the top of the list was disease modification. So in other words, actually making the joint to fix itself, okay, to be fixed. The bottom of the list was pain. Okay, because of course we can deal with pain, that's fine, wasn't it? You ask the patients, and the list is the other way up. Actually, they don't really care whether their joint is remodeling by different collagens forming this and forming that. What they care about is the pain and the malaise. Okay? The list is the other way up. And what you'll see increasingly, actually, and by the way, these are all, whoa. Ta-da. We're back, okay? Thank you so much. What you'll see increasingly is that pharmaceuticals are now engaging, and other researchers are now engaging very much more with patients, because that's really where we're getting the essence of where we need to target. But it's not quite so easy a story. There are these people involved, the regulators, and they're extremely important, because essentially what they will do is they will do a risk analysis and say, right, you've got this new medicine, okay? It's going to treat this disease. You've done the clinical trials. But you have these side effects. Now, do those side effects actually warrant the benefits that you're bringing with the medicine? So they're the regulators. And by the way, I promise not to speak on Brexit, but I will. One of the most fantastic things is a small amendment recently was that in the UK, we will continue to have the European regulations on our medicines. That is massive for research in the UK. A small amendment, but the fact that we will be using the same regulators is still absolutely important. What you then have in the way, if you like, is an organisation like NICE, which will decide whether there's a cost-benefit ratio, okay? And then you move on to the physician who will need to prescribe the medicine. Okay, so it's quite a complex route to get the pathway to the patient. But these have all been worked through with various other conditions, and we need to do the same with ME. And one of the things we were talking about having a meal last night is one of the most important things here is for us as researchers who may have early medicines is to know who to run a clinical trial on to test that medicine. Now, one of the most important things is to be able to have a clear set of diagnostic criteria to be able to do that. So one of the things I would be saying here is that work, as you described there, Professor Moreau, actually around how we can categorize the disease is going to be really critical to knowing where we target our medicines. Because if we go currently for ME, it's a very broad church. And actually for clinical trials, because they're in a limited set of people, we need a small group with a very clear phenotype, a very clear readout, a set of symptoms that we can measure and measure changes in. And actually the diagnostic element is, to me, as important as the cause and the effect element. One thing I also want to say here, and this is, I work quite a bit with Rare Disease UK, and that's a fantastic organisation. Actually looking at how we can use current medicines to actually hit some of the targets, or some of the symptoms, that are present in ME. So if I talked to my daughter and was to say to her, Elizabeth, I can't cure your ME with this medicine, but I can give you another 2 hours per day before you start getting chronic fatigue. What do you think about that? Yeah, please. Because that means that I can engage with schooling better, I can actually go out with my friends, I can actually even think of potentially getting some work. So those sort of activities, not just looking at what's the ultimate cure, but how can we hit symptoms, is going to be increasingly important. And the key point of this slide is this. This is the most important thing, the patient voice. Okay, one thing that The internet's given us, and very informed patients, is a very loud patient voice. And as I've said to you before, it has been said from this platform already, the patient advisory group is a really important element in that, in bringing that message forward to us. Finally, partnerships, and I've just got one slide on this. Partnerships are essential. You no longer see pharmaceuticals or any university or segment of the vendors or whatever actually just focusing on their own. It's all about partnership. We have great partnership with Oxford University and Harvard. Without that, we just would not be able to do the work we're doing. And you can see now, basically the way forward in research in pharma and academia is to blur those barriers and actually work together as units, as collaborations. Which is why the Research Collaborative is such an important element of what we're doing here in ME. So those are my 3 Ps. What I'm going to show you now is a video. We put this together essentially to show how essentially the whole research process of getting to a drug has developed over recent years. How much more it's about precision medicine. And the great thing is, my daughter Elizabeth is the patient at the beginning with ME. So hooray, we're getting it in there. Yes. Enjoy.
The dream of modern medicine is to move towards completely personalised healthcare, where each patient is treated as an individual. To get there, the essential ingredient is data science. Our journey starts with the patient.
I feel very fatigued and exhausted, and I get a lot of headaches and muscle ache. And the biggest symptom is post-exertion fatigue, where the minute you push your body more than it can take, you're then like way worse for the next week and in bed and stuff.
There are many people in the same situation as Elizabeth, with an unclear diagnosis. The cause of her illness is unknown, and there are no effective treatments.
If there was a drug out there that could help, it would just help people so much, because you'd know how long you were going to be ill for, and you could plan for the future, whereas we can't because we have no idea what's going to happen next.
One path is to explore how genetics may be driving the disease.
Fundamentally, we want to find a mutation in the patient's genome that is causing that individual's disease.
DNA samples are taken from patients and their parents, which generates a large amount of data.
So to begin with, Iwana will look for mutations that are not present in the parent. So this is a spontaneous mutation in the offspring. That is a likely mutation. It's more likely to have caused the disease that we see.
It can be quite difficult at times, yes, because we are basically looking for a needle in a haystack. We are looking at all these millions of bases in a person's genome, and we are trying to see what's different in that person and which might cause the severe disease they have.
The challenge for the data scientist is to sift through this information to find hints about what might cause disease.
So right now, the biggest advance in the genetic space is next generation sequencing. We can sequence the whole genome from an individual in less than a week for around about $1,000. So the ability to just generate that amount of data on an individual and family members so we understand allows us to pick out the genetics.
The ability to understand an individual patient like this is the origin of precision medicine.
Precision medicine requires the exquisite understanding of the biological processes that underlie disease. By understanding exactly how these processes work, how they are different from patient to patient, that allows us to actually adapt the treatment to the specific condition of each particular individual.
Once we have some leads from the genetics, the next challenge is to use information from every possible external source.
So this is UCB's Competitive Watch portal, and basically this represents an aggregated environment of externally published information which can include anything from scientific databases, patent indexes, clinical trial databases, and a whole raft of external public websites. Through this portal, the researcher can actually come and find real-time information that may be relevant for a particular disease that they may be working on or a particular project.
Jonathan Van El works through all of the biological data that's been published about the disease around the world to try to pull together all the research that's already been done.
Probably most of the data we use are coming from the external world, and I would say it's about 80% of the data we use. So our goal is not only to generate data or to make the genetics, but it's really to have those data sets to speak to each other, and the integration of those data will give new knowledge and new insights into the disease.
Using this vast array of external information helps guide and accelerate research.
This is really about getting the right information for the right decision point or for the right scientific research project at the right time, and that's what's really driving this, so that ultimately we can obviously do the best research we can to have the knock-on effect of delivering to the patients.
Once we have an idea of how the disease works, we can start designing drugs to treat it.
There are lots of different ways to deliver medicines to patients. UCB focuses on two specific approaches. One, which we refer to as small molecules, which is chemistry-based and generally is administered to a patient in pill form. The other we refer to as a large molecule. These are derived from antibodies, and generally we administer those through injection.
Ji Shi is helping understand the properties of large molecules. He uses 3D modelling software to make them virtually, and if they look promising, even makes models of the antibodies.
These models really help me to visualize the surface, the depth, and the complexity. And in particular, when you fit two proteins together, it gives us a very direct perspective of the surface properties of the molecule. In order to evaluate a broad spectrum of possibilities, we use the computational tools to help us narrow down, really zoom into the most probable configurations so that we can test and evaluate those limited number of configurations in the wet lab.
In the computer-aided drug design team in Belgium, Zara is designing small molecules.
So when I look at this molecule, I'm zooming in, I'm seeing what contacts this small molecule is forming with the protein, but also which contacts could it potentially pick up that it's not exploiting.
There are more potential drug-like molecules than there are stars in our universe. So you can imagine we need to have tools that can enable us to identify those very small needles in a very large haystack. Computers help you to focus on the areas that may be more fruitful, and they help you evaluate millions of ideas on a timescale that enables you to feed back to chemists so that they can then think about the molecules, how they're going to make them, and then we test them once they're made, and then we feed that information back into our models, and that helps us going forward.
After this computational design phase, it's time to test the drug candidates.
The standard process for evaluating a drug is to, first of all, think about how the drug will work in the disease setting, but also to ensure that it's a viable product that can be taken as a medicine. And all of this happens in model systems, in testing environments, before we ever take it anywhere near a healthy volunteer or a patient population.
3 or 4 years ago, if you looked at the experiments that were being performed for a project, 80-90% of those experiments were performed inside the walls of UCB, so the scientists on site performing those experiments. Now, it's completely reversed. It's actually only maybe 10 or 20% of the experiments are being performed inside the walls of UCB, and the vast amount of data is actually being generated, and most of the experiments are being performed outside.
Data is created by labs around the world and has to be brought back into UCB and made easier to interpret. Dan Smaragd is part of the team that does this vital job.
So here we're looking at data from an ongoing project at the moment. You can see data that have been captured by different systems and which are all aggregated together in this kind of visualization. So people can look at the trends and correlation between the data. You could say it's my job to make something complicated into something simple, yes.
So the complexity in understanding just the simple experiments is getting to a level where you need almost the same data analytics capability you had for an entire project 10 years ago is required to understand one of the experiments.
When we have a drug that looks promising, then come the clinical trials. Data science is essential to process all the results that are coming in and to understand what is happening in each individual patient.
Clinical trials are essential elements of the development of a drug. Before you can put a drug on the market, you need to make sure that the drug is safe and the drug is efficacious. And the only way to do that is actually to test that in patients.
Every time a patient comes in for a clinical trial, we measure RNA and DNA in its blood, and then what you see here is a different patient corresponding to different columns and genes to rows. So what we do is we use machine learning and data science algorithms, and we try to identify patterns in these fingerprints. And this will allow us to group patients together. Genes actually show us the disease mechanisms in every patient. Every patient has his own version or her own version of the disease. So by looking at the molecular fingerprint, we can see exactly what's wrong with every individual patient and helps us treat them better.
Irene is a statistician. She gets all the data from the trials and tries to find the patterns hidden in them. Ultimately, these data scientists are trying to understand what is going on inside each individual patient.
Hopefully what is going to change is that clinicians will be able to make decisions based on much more objective and precise measurements, which are biomarkers coming from this big data, and they will be able to give the right drug to the right patient in the first instance without having to go through that process of trial and error.
Medicine in the past, we were giving drugs essentially in a blanket fashion to all patients, whether they had particular markers that said that they would respond or not. Now we're sort of understanding different groups of patients, we can target therapies better. This will avoid, in our view, actually testing drugs in patients who may suffer side effects unnecessarily and gain no benefit.
In this journey, data science has allowed us to precisely understand disease and to filter and understand the vast amounts of data coming in from the drug development and testing process. It allows us to apply precision medicine to one group of patients, and then the journey starts all over again. The rapid growth of data and greater understanding of the causes of disease, there will be more and more opportunities for patients like Elizabeth in the future.
Our ultimate aim, of course, is to develop medicines that are truly personalised, where we can actually give a drug to a patient who has a particular molecular signature. However, it's a real journey, and I think we've probably only just started on it for most diseases.
Data science is essential for the future of medicine, not only for the part where we develop new medicines, but also for the way that medicine will be prescribed to the patients. So the physician himself will use data science tool to actually find the best prescription, the best treatment for a patient he has in front of him.
I think in the future we will have a lot more data, we'll have access to lots more different types of data, and where I see this going in the future is predictive algorithms rather than one-off individual tests based on one entity. It's going to be a combination of all this data together to understand patients and then to make sure we're placing the drugs and the treatments with the right patients.
There you go.
Oh, thank you very much.
Great.
That's wonderful.
So we need a lot of